Hello students, today I am going to discuss about chromosome walking. That is a very important technique of cytogenetics. With the help of this technique, different genes and markers on a chromosomal fragment can be identified and characterized. So basically the chromosome walking gives an idea about linear arrangement of genes and the different DNA markers, and also about their relative positions on the chromosome. So what is the requirement of chromosome walking? When a genome is characterized, suppose there is a DNA fragment. This is a DNA fragment, a large DNA fragment of chromosome. And when you want to characterize this for the presence of different genes like A, B, different markers C, D, E, F. So how you can find out the linear arrangement? A particular linear arrangement that A is coming before B, and similarly this is the linear arrangement. How this linear arrangement can be identified in genome sequencing projects? For this, uh, shotgun method is applied, in which this large DNA fragment is broken into smaller parts, and these smaller parts are characterized for their DNA sequence and different markers, and then a master sequence is prepared. So but the major limitation of sequencing technologies is that no method can sequence more than 750 base pair DNA fragment in a single experiment. So you need to adopt different strategies. So what are those strategies? Um, First of all, break the chromosomal fragment or the larger DNA into small part. So suppose this is the larger DNA. First you break this into smaller parts. Then generate a gene library. I have already taken a lecture on gene library. So you generate a gene library and prepare a plate where different colonies of bacteria is present, and these colonies are having different DNA fragments. So the bigger DNA fragment, then they are broken into smaller bit and the gene library is generated. Now each gene library is probed with a known DNA or marker or any restriction fragment to identify the presence of a particular DNA fragment in the library, and this is done repeatedly to identify different clones, and this also gives you the linear order of different genes and DNA marker on the original or parental chromosome. So now this is clear that the chromosome walking is used for characterizing long stretches of a chromosomal fragment. This technique can characterize a DNA fragment or chromosomal fragment of about 1000 kilobase per in length. So now the important point about this. First of all, suppose this is a chromosomal fragment that you want to characterize. And now you need to know any specific gene, any specific gene near this DNA fragment for the characterization. So a known gene or any known marker must be present before or after the chromosomal DNA fragment that you want to characterize. So this is the process starts with a known gene present near the gene of interest on DNA fragment. Okay, so now how this works? Uh, Different clones are obtained in genomic library. So first of all, you will create a genomic library from this DNA fragment. And now the chromosome walking experiment is done, right? So how that is done? The end piece of a cloned DNA fragment is subcloned and is used as a probe to recover another overlapping clone from the genomic library. What they are saying, this is a suppose original DNA fragment before fragmentation and before preparing the gene library, and now you know you have one known gene present at 5' end of this chromosomal fragment. This is known DNA. So you will prepare a probe. You will prepare a probe by subcloning subcloning one end of this known gene, and the probe will be made from a particular end of this gene. Now you will probe and you will identify, suppose there is one fragment in the gene library. So now you can, this probe will bind with this fragment region, and now, so you know that, and you will sequence the whole DNA fragment. Now you will know, suppose there is marker A, the first of all marker A is coming at 5 prime end. Similarly, now you have identified this clone, suppose the clone 1 you have identified, and now you will subclone the 3 prime end further for developing another probe. This was probe 1, now this is probe 2, and again you will probe the whole library for find out the overlapping clone that binds with P2. Say for example you got another clone and that is having overlapping region. So now you will sequence this and characterize this smaller fragment. Suppose you got B, C, here D. Now it is clear that this is the order A, B, C, D, and the process would be more clear in the next slide. Please go through each and every step one by one. I have already told you an overview of the process in my previous slide, and the pictorial presentation is given in next slide. Suppose this is the target DNA that you want to characterize, and this target DNA is flanked by two markers A and V. Now we will start chromosome walking in this direction. So you already know a molecular marker A present at 5 prime end of this target DNA, and you have already prepared a gene library from this DNA, right? So you will probe this library for the presence of different DNA fragment using a specific labeled probe. So the first probe that you will prepare would be the flanking region of the DNA fragment that is known. So now this is the probe, and you will hybridize the gene library. The process I have already told you. A membrane will be kept over the gene library. So now different colonies will be adhered onto membrane, and the DNA is lysed and fixed at their respective position on the membrane, and then this membrane is hybridized with the probe. So what would be different probes that I am telling you? The first probe is here. This is the first probe. This probe will identify any colony, say for example this colony, number 2 colony. So this means this colony is having a complementary region to this probe. And now you will isolate the DNA from this colony. You will isolate the DNA from this colony, and you will sequence it, and you will find the remaining region. So this fragment is characterized using the first probe P1. Now you will have the sequence of B DNA fragment, say for example, and now again you will subclone and prepare another probe P2 that is present at the 3 prime end of B fragment, and again you will probe the whole library with this probe, and you will identify, say for example, another fragment C, and now again you will use another probe P3 that is the flanking region of DNA fragment C. So this way you are seeing, using different probes, you are walking down the chromosome and you are characterizing different fragments. Suppose B, C, now this is D and E. So now at the end of this experiment you will have the full information, and you will keep on walking till you identify the marker B that is already known to you. So now you have characterized the DNA fragment between two marker A and B present on any chromosomal fragment. This is also the pictorial representation, as I have told you in my previous slide. This photograph is having colored markers present on any eukaryotic DNA, and the process is same. So kindly look at this to see that how different fragments having overlapping regions are characterized using different probes, which are nothing but the 3' end regions of these DNA fragment. And this is how the chromosome walking is done to characterize the whole fragment. In this case, a library is prepared in lambda phase and the chromosome walking is performed. Thank you. You may call me for any doubt.